Hi everyone, it's Mr. Sinti, and today we're going to talk about one of the classics of all time in biology, mitosis. Yes, and you will understand this and it'll be fabulous. And so I want to start off before I get too far into it by just sort of reviewing a little bit of the cell cycle from our previous discussion. Remember that a cell, when it's growing, moves along in a phase called interphase, and basically it's a in between. It's called inter because it's in between when it's actually dividing. So it's interphase, mitosis, interphase, mitosis, interphase, mitosis. And so as the cell goes along in interphase, its interphase is so long, 23 out of 24 hours, it's broken up into three parts. As you can see here, G1, S, and G2. We'll talk about this momentarily. Basically, the cell's growing, then it replicates its DNA because it's anticipating the fact that it's going to split into two daughter cells, and then it continues to grow, and then it gets too huge. Its volume is more than its surface area can sort of sustain. So it undergoes this hour process called mitosis, which is the focus of this conversation today. And then the last 5 minutes before it actually breaks up is called cytokinesis, which is cell division, where the cell pinches in into two cells. And so let me sort of, before we get into this, let's go back a little bit. The last time I was talking to you, it was a little bit of a cliffhanger, and I sort of want to get back to that point. Like, say you have a cell, and it's really huge, and it's anticipating that it needs to divide. And if you're following this, you'll know that this is at the end of interphase in a period called G2 or gap 2. Over here, let me make the nucleus over to one side. Here's the nucleus. And so you may know that if the cell contains DNA, let's go red with DNA. Now, prior to the cell dividing into two cells, during the S phase of interphase, the DNA replicated or DNA synthesis occurs. Well, I'm not going into the detail of this, though I could. We'll talk about that later. Do you notice how there's two copies here? And I was saying, well, it's very difficult to travel with chromatin. There's so many strands of DNA. It's 3 meters long. It's a big deal. And you're like, well, I remember we have to condense it into chromosomes. And so if I were to sort of erase the chromatin and say this and go, all right, so we're going to coil it up around histone proteins, and we're going to make, instead of making lots of different chromosomes, let me sort of just be funny and say that we have one large chromosome, like this. We have one large chromosome in the nucleus. Do you remember? Let me back that down a little bit. Let's say that one side is called a sister chromatid and the other side's a sister chromatid, and so this whole thing is a chromosome. And I said that what the cell now needs to do is to pull one sister chromatid over to the other side of the cell and this one to this side of the cell. But here's the problem. You're not getting anywhere when you have a nuclear membrane. So you ready for this? What we're going to be talking about today is this whole idea of mitosis. And let me just go right here and say mitosis. Mitosis is a preparatory process before a cell divides, and what it's all about, and it can be broken up into different phases, but just hold off on that for a second. What happens in mitosis is that the DNA, the duplicated DNA, coils up into chromosomes, but then you have to get rid of the nucleus. And you're like, can you do that? Yeah. The membrane can not disappear, but it can dissolve and become part of the Golgi or part of the plasma membrane. But you're not going to be able to move the chromosome with a nuclear membrane. I think that's obvious, but I just want to state that point. And then obviously, you're not going to get the chromosome to move either unless you pull on it. And so what's interesting is there's a center protein called the centromere. And then there's these other little proteins that come off, sort of like little hands off the sides. I'll name all this in a moment. And then there's proteins that are being produced by the corners of the cell. These are called centrioles. And these fibers come out, and they're going to grab the chromosome, and over on this side too. and they're going to pull on it and tug on the chromosome. And it's like, well, where do you want it? Where do you want it? Well, what's going to happen is, let me make the, let's see if this actually makes the eraser larger. Yeah, that's good. So what's going to happen is it's going to pull. Let me sort of move it over here. Now, obviously, there's more than one chromosome. So let me go 2 chromosomes here. So there's one chromosome, and then there's another chromosome. So they get pulled to the true middle of the cell. Now, like, why would you do that? Because this way, you can make sure that none of the chromosomes are going to get lost if you put them in the center of the cell, like this. And so you're going to grab them and push them to the center of the cell, and then you're going to like yank on them and just sort of pull them apart from one another. And so they're going to split like this, right into the center like that. And it's like, oh no, oh no. But all the cell needs is one side of the DNA, because both of these are identical to one another. And so difficult to animate this, but a little technical difficulty. I'm just going to roll with it. This is going to go to one side of the cell, and this is going to go to the other, and this is going to go to the other and the other. And so the idea is to pull the DNA apart, and then the cell's going to be good to go to divide. And so let's erase that. Let's go here. Let's go here. And so let's have at this. So what I want to talk about is, in earnest, is this mitosis, this last sort of hour of the cell cycle. But interphase, as I mentioned before, is really long, and it's 90% of the cell cycle. And what you can see here, it's sort of, this looks like a little bit of a cycle, but here's a typical cell in interphase. You can see the nucleus, you can see the DNA is here in blue. It sort of looks like a round nucleus, chromatin in the material, in the nucleus. And you're like, hey, what's going on? It looks kind of wormy here. Well, this is how the DNA is forming chromosomes, and then the nucleus breaks down. There's no more nucleus. Oh no, oh no. And so what happens is these little fibers pull on the DNA, the chromosomes, and line them up in the middle, and then they pull them apart, as I was showing a second ago. And once the DNA is now on opposite sides of the cell, this is still one cell, that's the end of mitosis. So mitosis is simply, you ready for this? The formation of chromosomes and the movement of DNA to opposite sides of the cell. You're like, well, why is that? Because the cell is about to divide, and when it divides, you don't want DNA on one side too much or one side too much on the other. So the new nucleus forms, and then cytokinesis occurs, and voila, you have 2 identical clone cells, asexual reproduction. And so that interphase is such a huge phase. I mentioned it's broken up into 3 parts. It's called G1, S, and G2. G doesn't, it could mean growth, but it doesn't mean growth. When the scientists first saw cells under the microscope, they sort of looked like they were sleeping, or there was some sort of like gap in between when the cell was doing something. It seemed like when they saw the chromosomes moving during mitosis, it was like, oh, cell's awake, cell's doing something. But in fact, the cell's doing more during interphase, because it's making protein, it's growing, it's copying its organelles, it's synthesizing its DNA. So you can see here it's growing, it's making proteins, it's replicating its DNA, it's getting really large. And then if you wanted to put in a detail here, during G2, it's actually making a lot of a protein called tubulin, because tubulin is going to be comprising the microtubule apparatus that forms the spindle that reaches out and grabs the chromosomes and pulls them apart. And so that's pretty cool. And then the daughter cells repeat, and so thus it's called a cycle. And so let's get into mitosis. And so, mitosis, I'm not sure if you asked a cell if it could talk, but if you asked it, like, what phase are you in, it wouldn't be able to answer this. It's just, obviously this is humans looking at the cell cycle and trying to partition it into little areas, like chapters of a book, and saying, this is what's happening. Now this is what's happening. Now this is what's happening. But mitosis is a continuous process. In other words, it's like taking a trip. You get your clothes, you wrap them up, you put them in the suitcase, you walk them to the door, you line them up so that you don't lose any, then you travel, then you get to the hotel, you unpack your luggage. It's a continuous flow. But we could break it up into different phases. And so biologists have done this. So here are the 4 phases of mitosis. The first phase is, guess what, is called prophase, which literally means the first phase. So there's a picture of classic prophase. It's just focusing in on the nucleus. You can see the chromosomes are forming. That's obviously has to go first. You've got to wrap up your DNA around the histone proteins. And then what's happening here is, if I stop advancing the slide, the nuclear membrane will start to break down, and then as you can see in the picture below, this is metaphase. A great name, meta meaning in the middle. So all the chromosomes are lined up in the true equator of the cell. Anaphase is the third phase, and if you will, this is where the sister chromatids are pulled apart and the DNA is actually being moved. You know how like in a CD sometimes there's a title of a song and it's the same title as the name of the CD, sometimes called the title track. If mitosis had a title track, it would be anaphase, because that's actually what mitosis is, the movement of DNA. But it's not just the movement of DNA, because you've got to form the chromosomes, got to get rid of the nucleus, got to line them up, and then you got to pull them apart. The word mitosis itself actually means threads. Again, a great name, because as you can see, all these spindle fiber threads are what's pulling the DNA apart. And then finally, telo means at the end. Telo means end. So it's the end phase. First phase, middle phase, pull apart phase, and at the end phase. Here's the cell. It's still one cell. The DNA has now arrived. A new nucleus is forming in both sides of the cytoplasm. The DNA is now decondensing or unraveling off of those proteins, forming chromatin again. And so now the cell is ready for cytokinesis. So mitosis is not cell division. I just want to emphasize that. Mitosis is not cell division. It is what occurs in the cell before the cell divides, because you don't want the DNA stuck on both sides or on the same side. You want to be able to partition it. So mitosis occurs before cell division. So another awesome picture here. What is it? As you can see, here's just the nucleus. This is the whole cell. These yellow threads are the microtubules of the cytoskeleton. At this point, heading into mitosis, the DNA is already replicated, but you can't see it. It's kind of loose. So all this chromatin is in there, and the nucleus sort of looks circular. And so what's happening is these spindles starting to radiate. In other words, these proteins are getting long. And when scientists first saw that under the microscope, it sort of reminded them of a sun that was sort of somewhat poetic. It's like a sun that's radiating like this. And as you may know, that this is the symbol of an asterex, or an aster meaning star, or the study of astronomy. So it's kind of like these stars are emanating out from the sides. Now, you're like, they're not just coming out of nowhere. There's 2 centrioles that are doing this. But what's interesting is just prior to mitosis, the centrioles, there's 2 of them normally in the cell. They actually replicate. So now there's 4 of them. And you're like, oh no, 4 of them. So then these 2 are going to giddy up over to the other side of the cell so that they can actually start producing spindle fibers in this direction so we can move the DNA. Maybe a better picture coming. So this sort of looks like one of those stuffed toys called a troll. I don't know what the eyes here, I don't know. But here are the 2 centrioles here. And what's happening is you can see the chromosomes are starting to form. So this is early prophase. The nucleus still looks intact, so you're not going to be able to do anything. So prophase, the chromosomes are being formed. So that's very important. You're like, what's prophase? It's the first phase. What's going to go on in mitosis? The DNA coils itself up around histone proteins and chromosomes form. That's the big deal. The nuclear membrane is going to need to disappear too, because we've talked about this, because with the nuclear membranes there, you're not going to be able to move the chromosomes. So here's a little review of how DNA forms a chromosome. It hooks up around these histone proteins and it condenses, forming this big nucleosome complex, and then further supercoiling, and then whoa, you have this big X-shaped structure. This side is identical to this side. So the point is, you've got to separate sister chromatids during mitosis before division. So there it is. This side up here is the same as this side down here. As you can see, it gets thicker and thicker until you see two sister chromatids that make up one chromosome. Chromosome means colored body. Kind of cool. Now I mentioned this before. The centromere is a protein in the center of the chromosome that holds the sisters together. But remember I mentioned before there's like these little hand proteins that come off the side? These proteins, it seems like a detail and you may not want to know this, but let me assure you this is actually fairly significant. These little hands that are coming off, let me emphasize it in blue. These little protein fibers that are coming off the sides here are called kinetochore. They're also made up of proteins. And these are significant because those yellow spindle fibers are going to come and sort of handshake these proteins. And this is what's actually going to pull the chromosome apart from one another. And let me sort of just take a break from this and show you what I mean by that, if I can. I'm not sure if I'm going to be able to, but let's see if we can do this. Let's go. struggle. Let's go over here. In this animation, you can see what's happening. If I sort of tell you, this in red is the chromosome, and this green are the spindle fibers that are, and you can see how they, how they're actually attached to the chromosomes. It's kind of brief. This might be a little bit more interesting, though it's not in color. It's okay. Here's the whole cell, and here if I outline it, you're like, ooh, a dark region. This is the nucleus of the cell. Here's the cell membrane. You're like, ooh, this area is glowing. Under the microscope, we were able to label tubulin proteins. So you can see over here the centrioles are really glowing. You can see like a star, the asterisk, spindle fibers are forming. But again, nothing inside because the proteins cannot penetrate in here because this is the nucleus. You've got to get rid of the nuclear membrane if you're going to move the chromosomes around. So let me just let this play out. And so notice here the nucleus disappeared, and ooh, look, now the spindle fibers are actually able to connect. Now you're not going to see the DNA. It's slightly disappointing, but there'd be chromosomes in the center here. But this is only emphasizing the spindle fibers. But there's chromosomes in the center. Let me let it play. They're grabbing along the DNA, and then they depolymerize. They're going to pull the DNA apart from one another. So that's kind of a cool animation, I think. Let's go back over here, maybe, and perhaps. You can see here, this is sort of at the end of prophase. Sometimes, depending on the level of your study, you could say that there's not four phases like pro, meta, anatella. There's actually something called prometaphase because prophase, sure, the nucleus breaks down, sure, the chromosomes form, but then there's sort of a period there where the chromosomes have to be sort of dragged to the middle, and that's called prometaphase. And so just wanted to point that out. So the nuclear envelope breaks down, the microtubules sort of grab a hold of the kinetochore fibers of the chromosome and start yanking them to the true middle of the cell, although it's not shown that way, but that's exactly what's happening. Kinetochores are those proteins on the end. So this is a great shot. This shows the spindle apparatus. It shows the nucleus. That was that dark spot in the video previously. Here's the nuclear membrane breaking down right there. And so here's another picture of the cell using fluorescence. The chromosomes are in blue, the spindle are in yellow. Notice how at this point there's no nucleus at all. And how do I know that? Because the chromosomes are in the true middle of the cell. They wouldn't be able to get to the middle of the cell if there was a nucleus because I was holding them back. So they're right in the true middle, or what's called the metaphase plate. So they line up. And there they are all lining up in the center right there. That's kind of cool. And so the next thing that's going to happen after metaphase is anaphase, and that's the actual separating of sister chromatids. And so here is the spindle fibers, sort of like a fisherman hooking the chromosome or a fish and reeling them in. So the DNA is literally being pulled to the left or pulled to the right, or if this is, it's obviously 3D, if this is going to the top, this is going to the bottom. So what's the point? The point is you've got to separate the DNA because the cell is about to divide. So anaphase separates the sister chromatids. Very important. And so they're being pulled apart. And so the equivalent amount of DNA is moving left and right. And so this is a beautiful shot. And I like this shot because it shows many cells. So this is a tissue. I don't know if you thought about this, but not every cell is undergoing mitosis at the same time. The cells are sort of doing their own thing. They're like, in other words, not everyone at our school is the same age. They're at different stages of their life. So these are interphase, 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 and you're like, whoa, gee, a lot of cells are in interphase. It's predictable that there would be more interphase cells because it takes so much longer to be in interphase. Mitosis only takes an hour, so it's sort of somewhat fleeting and it's rare. So here it is. Notice how it's one cell. Before the cell can divide, the sister chromatids must be pulled apart. Beautiful shot of anaphase, which is the third phase of mitosis. Close-up of one chromosome that's being pulled apart. Here's one sister going to one side, the other sister going to the other side, and what's happening? The spindle fibers are doing it. Now, let me back this off for a second and show you, if I can, another animation, if this will let me do it. I'm not sure if it'll let me do it. Sorry. Let's go. Nah, it's not gonna. Doesn't like me now. Here. Let me try this one more time. All right, stop it over here. Let me close that down. Okay, this is what I want. Sorry. This is a time lapse because mitosis takes an hour. So this is a time lapse of mitosis, and I think it's appropriate to sort of like pause here and take a look at this. Now, you can't see this. I don't see it either, but this is approximately like what the cell's looking like. Here's a cell membrane. You're like, oh yeah. All right, so what's happening here? This is the nuclear membrane. And what's happening? I'd say if I were to ask you what phase of the cell cycle this is, you would say, well, since there's a nucleus, it's got to be either interphase or the beginning of prophase. Do you get it? Because this is what, when there's a nucleus, there's no nucleus during metaphase, no nucleus during anaphase, and then there'll be two nuclei forming during telophase on both sides of the cell. So I'm gonna call this out saying this is towards the end of interphase because I'm starting to see little wormies forming in the center here. So it's kind of like chromosomes. Now, this is a video, and so let me let it play out and I'll sort of talk about what's happening. Look at the nuclear membrane. You know, remember one of the first things that happens in mitosis prophase is the nuclear membrane goes away. So there it is going away, and then chromosomes form. So this is prophase. Now, what's happening is these chromosomes need to get to the center of the cell. So what's happening is the spindle fibers are grabbing them and pulling them. So this is like prometaphase. So they're being yanked and pulled and pulled. And they're like, get to the center of the cell. And so now there they are in the true center of the cell. I would call this metaphase. And now, if you anticipate what's going to happen next, it's going to be anaphase. And you're going to see the sisters being yanked apart in a moment. So now the spindle fibers pull on the sister chromatids, and so that's a beautiful picture of anaphase. And you're like, well, what comes next? Telophase or telophase. Notice how the sisters are still condensed, but like just check out what happens to the chromatid when it arrives. So you get to the hotel. First thing that you got to do is uncoil. So do you notice how two nuclei are forming? I can see them right here, and then the DNA is looking kind of loose again. This is what you'd call telophase. So it's sort of the opposite of prophase. Nucleus forms, DNA decondenses. That's the end of mitosis. There it is. Those are the four phases. And you're like, well, the cell has not divided. Cell division is called cytokinesis, and as you can see here, the membrane is pinching in. You're like, wow, membrane's pinching in. How can it happen? Well, it's a phospholipid bilayer. It's fluid. It can pinch in. Who's pulling the membrane in? Microfilaments. those are made up of a protein called actin, and those are what's pulling it in. So check it out. Here's cytokinesis, or cell division. Whoa. And then now you have two identical daughter cells. There you go. The cell cycle. That's pretty cool, huh? I like that. Sometimes that's more informative than sort of looking at it step by step. But the step by step is important too, because then you can talk about it more easily. So there's anaphase. There's a nice picture of anaphase there. And then when the chromatids arrive, they start to decondense. The cell elongates. Two nuclei are starting to form, and so telophase or telophase. The DNA is decondensing into chromatin, and the cell is looking to divide. So telophase is the end of mitosis, and then cytokinesis is cell division. So just in summary of mitosis, here's a cell in interphase. You can see how it has a nice nucleus. DNA is loose, called chromatin. During interphase, the DNA replicates, even though you can't see that. Then it condenses into chromosomes during prophase. The spindle starts to form, nuclear membranes breaking down, oh my goodness. And then here's that picture of prometaphase, where the spindles are yanking the chromosomes to the true middle. In this creature has 4 chromosomes. Now we have 46, just saying. So this creature has 4, that's fine. It's easier to follow. And so the sister chromatids get pulled apart, 4 go this way, 4 go that way, and that's all you really want, because these are identical. And then, ooh, guess what? The DNA arrives, it decondenses, a new nucleus forms, we saw that, and then the cell actually divides, which is called cytokinesis. And so this is a picture of, if you would guess it, you can still see the nucleus, prophase. Let me go back, that's prophase. Here is metaphase, all the chromosomes are in the middle. This is a plant cell, you can see how there's a cell wall. Here's a cool picture, also a plant cell of anaphase. Chromosomes are being pulled apart. Sister chromatids are going to opposite sides of the cell. And then here's a great picture of telophase, the last phase. And you're like, well, you might ask this question. If you're a plant cell and you're trying to divide, you're in a little bit more trouble, because the membrane can't really pinch in, or the membrane can pinch in, but the cell wall isn't going to be able to pinch in. Let me do a different color for the wall. So see how the wall is really rigid like this? You're like, well, now what are you going to do? Well, what has to happen is a new cellulose wall, you can even see it forming, a new cellulose wall starts to form. It's called a cell plate. And when that cell wall forms, then the membrane can simply form around like this and like that. And you got, hey, 2 new cells. So the cell membrane doesn't quite pinch in like it does in an animal cell. That's fine. Although this is what a cell looks like when it's pinching in. You know, it is 3D, and so this is actually 4 cells. And it's called cytokinesis. And when it pinches in like this, it forms what scientists call sort of this pinching in called a cleavage furrow. And so this is the indentation that occurs when the cells are dividing, and that's being pulled in by the thin filaments, the actin filaments. So you can actually see it here right there. That's kind of cool. The 2 daughter cells are identical. And then, you know that picture that I was drawing a second ago? This is the actual cell plate again. So that forms. First, little membrane starts to form on one side and the other out of like Golgi apparatus, and then a cell wall is laid down in the center. And so that allows the plant cell to be able to divide into 2. And then in summary, we're getting to the end here. Here's interphase, prometa, metaphase, anaphase, telophase, and then cytokinesis. And then finally, I wanted to point out, since bacteria are indeed the most numerous organisms on the earth, they only live for about 30 minutes. And it's like they don't have to go through the big mitosis, and they don't need to get rid of the nucleus. And you're like, well, they don't even have a nucleus. And so what are they going to do? And so all they need to do, it's kind of cool, and I have to emphasize it. What they need to do is, when they have their DNA, what they do is they replicate their DNA, and then they hook it to the cell membrane, sort of like a piece of clothing stuck to a clothesline with a clothespin. And then the other piece of DNA, they hook it to the cell membrane. And so when the cell membrane moves, like this, the 2 DNAs move far enough apart, and then the cell divides. So they don't have to go through the big mitosis thing. So when they reproduce, it's called binary, which means two, binary fission. It's not mitosis, and they do this every 30 minutes in ideal conditions. And so they don't have as much DNA. They only have one piece. They have one chromosome. They don't have 46 chromosomes. And so here's an example of E. coli. It's got one chromosome, and then you've got to replicate it. So then, oh, look, you have two pieces of DNA. And then, as I mentioned before, you hook it to the cell membrane. And then as the cell gets longer in size, gets larger, then the cell pinches in two, and you're good. You've divided into two new cells. So I hope you enjoyed our conversation of the cell cycle and emphasizing mitosis. It's a classic. I hope you enjoyed it. Thanks for watching.